Hello, my name is Daniela.
Hi, my name is Felicia.
Hi, my name is Nuri.
Practical 1. Spectrophotometry. The objective of this experiment is to introduce the students to spectrophotometry for qualitative analysis and quantitative analysis of biological samples and molecules. These were the materials used in the experiment. Spectrophotometer was used to measure how much light the sample absorbs to determine the protein concentration. The cuvette holds the sample inside the spectrophotometer. The Bradford reagent turns blue when it binds to protein, so the color intensity tells us how much protein is present. Distilled water was used to prepare sample dilutions. Test tubes and test tube rack were used to hold and organize the samples during preparation. Micropipettes were used to accurately measure and transfer small liquid volumes. The 70% ethanol solution was used to clean the cuvettes to avoid contamination. The BSA protein was used to make a standard curve, while the standard unknown protein solutions A, B, and C were the samples of protein concentrations we measured in the experiment.
Part 1. Place 4 ml of distilled water in 6 test tubes using micropipette. Place 1 ml of Bradford reagent into each tube. Add in 50 microliter of BSA protein in test tube 1 and 2, 100 microliter in test tube 3 and 4, 200 microliter in test tube 5 and 6. Test tubes were shaken well. The solution in the test tubes were transferred into the cuvettes. The reading of a solvent were tested and recorded using spectrophotometer. Part 2. Place 4 ml of distilled water in 6 test tubes using micropipettes. Place 1 ml of Bradford reagent into each tube. Add in 100 microliter of standard unknown protein A in test tube 1 and 2, unknown standard protein B in test tube 3 and 4, unknown standard protein in test tube 5 and 6. Test tubes were shaken well. The solution in the test tubes were transferred into the cuvettes. The reading of the solvents were tested and recorded using spectrophotometer. In part 1 of the experiment, the absorbent readings show a general increase as the concentration increased, although the blank produced an unusual high absorbance that disrupts the normal linear trend. This suggests a possible baseline or preparation error, but the low standard deviations indicate that the replicate was still inconsistent. In part 2, the standard protein curve displays a clear and reliable linear relationship between protein concentration and absorbance, confirming that the assay performed correctly. By comparing the absorbance of sample A, B, and C to the standard curve, we determined that all 3 samples contain low protein concentration with only slight differences between them. Overall, the result shows that the protein assay in part 2 was accurate, while Part 1 highlights the importance of proper blank preparation for a smoother calibration curve.
For the conclusion of this experiment, spectrophotometry was used to measure protein concentration. A standard curve was generated from BSA standards showing a linear relationship between absorbance and concentration. This curve was then used to determine the concentrations of the unknown protein samples. The experiment successfully demonstrated how absorbance data can be used for accurate quantitative analysis and reinforce essential skills in pipetting, blanking the instrument, and interpreting spectrophotometric results.
From the practical, we learned to develop a strong understanding of how spectrophotometry is used to quantify proteins. We also learned how to prepare standards, operate the spectrophotometer, and interpret a standard curve. It also helps connect theoretical concepts like the Beer-Lambert law to real laboratory work. Unfortunately, there is a challenge that we face during doing the practical, where the air bubbles in pipette tips can lead to incorrect volumes being transferred and cause inconsistencies in our standard and sample preparations. But we managed to improve techniques through practice, where there is a noticeable improvements in accuracy. We successfully constructed a clear linear standard curve and used it to determine the concentrations of the unknown samples. During the practical, we also divided tasks efficiently, such as pipetting, mixing, measuring, and recording, which kept the workflow smooth and organized. Group members supported each other, asked questions, and offered help when needed.